Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PIcc Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PIcc is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
No problem. Welcome everyone, July 31st, 2024. We have our steering committee meeting. Thank you everyone for joining. There's a lot of updates, so I want to get started right away, and hopefully we have some time for discussion of the various topics. We start with some of the updates from the FDA, and I think, as always, Noor will share the PDFs and or links with you in the chat, and I believe we will have a website update as soon as possible. The first part is from CDRH, a 2022 to 25 strategic priority document that also mentions collaborative communities, which I just wanted to share. There's many new guidance documents and a few interesting elements that, again, we're always looking also on the drug side, because usually in some way, shape, or form, those rules trickle over into the device side. And one of these guidance documents is addressing misinformation, and in this case about medical devices and prescription drugs. So that is illustrating that synergy that I just mentioned. And some of the definitions in the guidance documents are very helpful. So that was shared, so a quick pointer here. The second guidance here is a so-called diversity action plan to improve enrollment and participants from underrepresented populations, minorities, etc. So this could also fit into our diversity, equity, and inclusion section, but I wanted to include it here because, to my knowledge, for the first time, including the appropriate references and cross-references, the enrollment goals and their disaggregation requirements are provided here. And it is, I think it's very helpful if you're working in this space to know this. The other part, which is in this case a draft guidance, as you see here, that came out in May, is in this case for laboratory manufacturers, and in this case here about IVD devices for immediate public health response in the absence of declaration, section 564. And why I wanted to mention this specifically is they give an example, for a PCR-based test for an infectious disease, generally recommends the following validation studies. And there is a concrete statement here about number of positives, negatives used. So this obviously represents what was generally required under emergency use authorization. But I just wanted to provide this as potential early evidence that very concrete elements are provided in these draft guidances. Of course, this is still open for commenting, so that might be helpful. There was in July, so this just came out, a guidance document about application user fees for combination products, which is a very unique element. The new user fees for Medufa and Pedufa are, I think, still pending, but I have some preliminary numbers for you in a second. But I wanted to provide this here that you can see that depending on what type of combination product you're using and which act is applied, this is listed among many other things in this guidance document. One new program, I think it is a pilot currently, is called the Guidance Snapshot. Now, the Guidance Snapshot is something that is probably best explained by taking a look at it, which is for certain guidances, the demand of the public to showcase what that looks like and explaining it. So they have a podcast section. You see here, this is distributed via Apple Podcasts, and then, the guidance document itself is linked. And there have been already a lot of pharmacokinetics, adjustments of variates. There's an ethical consideration for the clinical investigation of products involving children. So please take a look. So this is a pilot. I don't think it's available for every single guidance. And one of the limitations that they list here, just to show you quickly, is that this is for increase of general public awareness, and they're not a substitute for the actual guidance. That's just an acknowledged limitation. And the guidance snapshots also have a little bit of a more, let's call it patient-friendly summary type description. So I think that's a very interesting program that was just started a few days ago in July, but it covers, again, some of the key, very frequently touched guidance documents. And of course, there's a connect and an email on the bottom. So if you want to provide feedback on that, that would probably be appreciated. Here's one tool that I definitely want to recommend to everyone, which comes from OSIL, and it's called Valid Path. So, the name here has nothing to do with the Valid Act. No pun intended here. But it's a whole slide imaging processing and machine learning performance assessment tool. In my opinion, a very great start, 3 different functions. And I have already requested whether we can book a separate session to go over this particular tool and what it can and cannot do in a separate webinar. But until then, please take a look. And this tool is part of the catalog of regulatory science tools. And as you know, there are many of these tools in radiology, etc. But again, a quick pointer. And I hope that we can get the FDA internal developer to give a little talk or webinar around this to see a little bit more. And there was earlier in July a webinar on IVD product classification. If you've missed it, the webinar materials, the slides are available, and there's also a recording of the CDRH webinar available on YouTube, and the link is in the slide deck that we will share with you. But I thought that's a very important, distinction here regarding regulatory control, pre-market review pathways, and then low, moderate, and high-risk devices. One release that was, I think here it says attributed to Jeff Shurn, is from July 15, which is an increased or the reorganization for increased organizational agility and advanced public health mission between different parts of the FDA, including probably part of this was the strategic priority plan, as well as to ensure the efforts will meet what Medufa is proposing. I just wanted to mention this because it is directly tied to the strategic priority plan, which I think was done by the same group. One additional element here is, yes, that's correct. I think Jeff Shurn has officially left, I believe, already. And I don't think whether that's official or not, I can't disclose, but I think I heard that Michelle Tarver, who was instrumental in, oh yeah, I just see that in the chat. Wonderful. Yeah, Michelle Tarver, I think, is acting now in the same function. She's, of course, I hope, still a good friend of PIcc, so definitely an interesting development. This new element here is a so-called hub for rare diseases and advanced outcomes for patients. You see there's CBER and CEDAR participation, and the Accelerating Rare Disease Cures Act program is also included here. And this is a strategic coalition part of the hub. I'm not quite sure what that will look like. The information currently available is mostly providing the context, meaning in intra-divisional links. But I hope that this will cover also at least some of the device components. As promised, the currently available MEDUFA changes, I put them together here in this table. And the only reliable numbers that I was able to get so this is sorted in gray, the current, and in black, the future for 510(k) pre-market approval and de novo systems. And here, requests. And here are the standard fees, and here are the small business fees. And to my knowledge, the waivers apply for total gross receipts after approved as an SBD of say less than 30 million, and after approval of an SBD of 100 million or less would be the small business fees. So I just wanted to illustrate this because a lot of laboratories ask how much is a de novo or a PMA if the LDT rule continues to go through, and this seems to be that rate. A quick comparison here across the board shows that these user fees show a 12% increase, so that would be factor of 1.12. So these 12% are tied to several of the priorities that I just shared, and presumably some of these initiatives that are outlined in the prior slides. I don't know if there's questions about it. Last time I checked, these numbers were not yet available, so I think that is still being discussed. And great comment in the chat here. I'll let you read that for yourself. Other quick DC updates. I presume everyone has heard about it, that the Chevron rule has been overturned, interpreted right now as decreasing the power in terms of rulemaking of agencies, but that, of course, depends on the context. But I just wanted to share this as a document because it's one of those very, very interesting generic statements by the Supreme Court that might have far-reaching consequences. The second element here is an official letter that was signed by Bill Cassidy talking about whether or not under Chevron there is still, let's say, a vector pointing to the FDA really being able to regulate LDTs. Of course, this is a letter that was sent to us by many folks, so I wanted to share this. Whether this holds up, similar to a lawsuit, remains to be determined. As far as I know, nothing of this is by any means binding, but those are just forces, let's just call it, to be recognized. A similar open letter that went to Dr. Shuren that was signed by 4 members of Congress was related to something that we discussed here before, which is the so-called AI assurance laboratories, initially proposed in a publication coming from several different people, among them representatives of the FDA, I think Mayo Platform, I think it was a JAMA paper that proposed national AI assurance laboratories. And here in this letter, there was a potential conflict of interest pointed out. I'll let you read this. I just wanted to share this because there's such an enormous interest to regulate AI. And these assurance laboratories, maybe the word center would be better because those weren't really laboratories in a clear sense, is one element. But there's 8 questions posed. Sorry, I didn't paste all of this here, but there's very concrete questions proposed here. So if you're interested in AI as well as how to assure that that works, these are parallel streams. So in parallel to FDA regulation of LDTs and clear and potential clear modernization or additional amendments that have been proposed by some parties. This is a completely separate track, and this is not specific to pathology or diagnostics, but more general sort of interdisciplinary. One other factor here related to the LDT part that I'm very thankful that people have shared came from the House Appropriations Committee. I think it was July 15th, and you see here in this section, this committee recommends to the FDA to not regulate LDTs. Of course, this is also, to my knowledge, it might be weighing in heavily in the discussion, but I'm not sure whether this is legally binding. Maybe I can just open it up because I know there are a lot of people on this call that are very knowledgeable about this let's call it legal intricacies. I don't know if anyone wants to make any comments because it's been 2 weeks since this came out.
I hate to burst your bubble, but it is not legally binding.
It is not legally binding?
No, it is not.
Okay. Yeah, I heard something similar. I wasn't quite sure, but I think it counts sort of like a recommendation, right?
It's a strongly worded opinion that FDA is perfectly free to ignore.
At the FDA, what?
It's perfectly free to ignore.
Hopefully they won't.
Yeah. Similar discussion here. This is a JAMA forum article written by Scott Gottlieb. Basically also a letter to Congress. So what I tried to put together here in these last 4 or 5 slides are, I would call this different vectors that either point to Congress or come from Congress pointing to the agencies. I think with a common theme that the complexity of our diagnostic tools have surpassed current approaches. And I'm not saying that the FDA is the right group or that CLIA is the right element, but I just wanted to share all these components that were shared with us around the same theme that, the quality of information data training sets for SI is essential. So I think you get hopefully the common drift, and we wanted to share all these elements with you to ensure that you're well informed and that you know what's going on. HHS has released end of June, that just came out before our last meeting, the finalized rule of establishing so-called disincentives for the information blocking rule. I think by now most healthcare centers have implemented means and approaches for sharing information immediately, as well as some of the exemptions, but this is a noteworthy release from HHS. Finally, this is funding, I believe, if I recall correctly, on the order of $140 million from the National Science Foundation to establish 7 national AI intelligence or AI research institutes. And you see the 7 institutes here. One is on law and society, one on cyber threat intelligence, climate-land interaction and trade, adaptation, economy, climate as an acronym, then artificial and natural intelligence, ARNI, one about societal decision-making or AISDM, and then one about inclusive intelligent technology for education. So this is invite, and this is very closely focused on learning and how to integrate AI technologies into a classroom. I meant to highlight this in a different color because I thought that was a very important point. There was a great New York Times article a few months ago about that's needed. And then lastly here, the for exceptional education, tailored intervention, understanding of childhood development, et cetera, et cetera. Now, the two things why I wanted to point this out is that more funding through NSF is expected, and that these currently do not have medicine in their action. So these are, of course, sector spanning funding, but no focused AI research institute yet. But it's expected that there's probably many additional ones coming. I want to hand it over briefly to Noor if your audio works. Can I
Hi
hand it over?
everyone. Yeah, sorry, I was having some technical difficulties earlier. I hope everyone is doing well. So just a few updates on MDIC's side. We have officially released the agenda for our annual public forum, which is occurring September 10th and 11th of this year. It'll be happening in Washington, D.C. I just pasted the links for the APF. Joe, if you don't mind switching to the next slide. Yeah, that's just register now. Do you have a picture of the agenda as well?
Yes, I do. I have.
Yes, perfect.
Give me 1 second. Yep.
Thank you. So yes, it'll be 2 full days, and Dr. Robert Califf is one of our key speakers for this event, and we have a very impressive lineup for both days actually. So we're excited to have everyone there, and we hope to see some familiar faces from this group as well. And Joe, if you don't mind switching to the next slide. We also have our annual patient summit, which is a virtual event. It will not be occurring in person. And it's insights on patient science tools and evidence generation. So there might be some overlap with this group as well. That'll be occurring October 29th. And it's from 10 a.m. till 1:15 Eastern time. And the link for that is also in the chat. Feel free to register if you're interested in joining. And that's it from MDIC side. Thank you, Joe.
Oh, perfect. Thank you. I wanted to hand it over to Robert Tessier. Are you available, Bob, to give us a quick update or pointers from PNPL?
Sure, I'd be more than happy to. We happen to have a research group called the Pathology Assistance Focus Group as part of our organization, and we're constantly looking for them to provide insights on what they're doing in different areas. And specifically, one of the members of that particular group just produced a presentation for our group about process improvements in the gross room. And we're getting this out to a number of different folks, and including sharing it with the American Association of Pathologist Assistants, and very anxious to continue to share information that's being done in that particular area and other similar studies.
Perfect. Thank you. And I know you've also been very active on the pricing of digital pathology slides and working on that project, correct?
Yeah, let me also say that we continue to struggle with trying to determine exactly what the costs are for digital pathology slides. We've been meeting with a number of academic institutions as well as a large community hospital operations and smaller laboratories. And we have been gathering slowly but surely some costing information. The most we're able to come up with at this point is an overall number of a cost per slide. And we continue to feel that we're going to be able to get more detailed information. We anticipate that we'll have some good budgeting numbers to be able to offer by the end of the year. And to the extent we'll have that information available, we'll be more than happy to share it with the PICC group.
Thank you. That's amazing. And you know, the slides here and these topics of interest for the members of PNPL, thank you for sharing that. And you know, this falls currently into the element of pre-analytic variability, but it's very important to keep that in mind as well. Want to quickly jump over to a few elements regarding diagnostic circulating tumor DNA. This is a paper by Jurdi, by the lab of Adam Jurdi, demonstrating perioperative tumor-informed ctDNA detection and the relevance here for survival outcomes. And then the second one here is a pointer to a blog that I hope many of you know already. This is done by Bruce Quinn, pathologist and visionary, I might say. He has a blog called Discoveries in Health Policy, and I think Noor has already posted this in the chat, I hope. And he recently published a white paper on residual or minimal residual disease testing, a case study for policy changes. And you see very concrete numbers as well as definitions in here. And just in general, I think it's a great blog to stay focused on those topics. One quick pointer to an excellent resource from the National Security Agency around cybersecurity. This was shared with us by one of the people from within the agency as a resource. There's quite a few elements. The final Zero Trust Pillar report that outlines how to discuss threat response might be helpful if you have witnessed the CrowdStrike incident and what to do there. And there's a lot of software memory safety reports. Those have actually been published before that incident. And just to be aware, these are quite technical assessments and recommendations. And the cybersecurity information sheet, I think, will be also on our website update. The diversity, equity, and inclusion part, I deferred to the earlier section within the FDA part. And one quick update here is from the Friends of Cancer Research project. Let me just see, I think Brittany is on. This here was a FDA reporting summary. I don't know, Brittany, if you want to make a few notes about this and or the digital pathology project, if you have some minutes time.
Yeah, sure. So this was, required under PDUFA, and FDA had put out the submissions that reported using real-world evidence in their submissions. And so for Friends, we just compiled this information into a few graphs to make it a little bit easier on the eyes than going through kind of all the tables that are listed on FDA's website. And so to be used as a resource for stakeholders to see where there's current interest in being able to apply real-world evidence. And then regarding our digital pathology project, I think the last update I had provided this group as a reminder for looking through concordance across 10 models, assessing HER2 and over 1,000 breast cancer whole slide images. We had previously been looking at the level of agreement for the ASCO CAP scores. We actually now just have data that we'll be discussing with our working group next week that's looking at the more quantitative H scores as well as the percent of tumor cells identified within the different ASCO CAP categories to get a little bit into the quantitative piece for continuous scoring and concordance there. So more data to come, and we're really excited to be able to share that more broadly with the community soon, all again with the goal of being able to help support alignment and use of these tools.
It's really amazing. I think that project is already extremely informative, and thank you for spearheading and taking this on, especially in light of HER2 low and other antibody drug conjugates. So that I think the project will be a blueprint how to do such things and model comparison, etc. Thank you, Brittany, for the quick update. A few updates from Europe. I think you may have seen that early in June, the EU regulation on AI has been finalized. And there was one interesting press release from the WHO that I also want to highlight quickly, end of June. I will show the paper in a second. This is results emphasized by IARC researchers about a low-cost battery-powered device for thermal ablation of cervical cancers and or precancers. And get to that in a second, but I just wanted to point out that that was emphasized as one of the great breakthroughs and how to apply certain technologies in low economic infrastructure. Then this is a joint statement from CAMD and HMA. So this is the heads of the medicine agencies. So this is the various, EMA bodies and their representatives, as well as the so-called competent authorities for medical devices. So these are large groups. They came together and provided a so-called statement about the regulations around the EU regulatory system for medical device regulation under IVDR. And they acknowledge that those regulations are to improve confidence and provide a solid basis, of course. And they call on the EU Commission to clearly prioritize medical devices in their next mandate, because there's a discrepancy between what ultimate test or IVDR developers, the health authorities, and the EU wants. So this is basically an indication that there's still a little bit of uncertainty in that middle layer between top down and bottom up on how to actually regulate this. And this group has effectively asked for help in terms of prioritization, which is I'm just highlighting two elements of a multi-page letter. So if you're interested in that topic, please take a look. We have received an astounding amount of resources this last month, probably due to the longer break because of the June, July, and or maybe the vacation times, but phenomenal resources. So I want to quickly go through this. Staying with the EU theme, a lot of people ask how do you manage a study prospective, potentially in pathology, obtaining data that are GDPR compliant, or how to organize this. And here's an example of a UK-based AI study. I believe a prostate AI algorithm is used, and the Articulate Pro study is the name. And they link back to NHS privacy notification system as well as consenting. So just wanted to point that out. So if you want to take a look at that's definitely possible. Another phenomenal resource is this paper by Sir Austin Bradford Hill, explaining the difference between association and causation on the topic of environment and disease. I thought that's a great read. Wanted to share this, of course. One paper that was provided, which is by David Blumke, which is coming from radiology as a quick overview of what to look for authors, reviewers, and readers, of course, of that journal. But I thought the key considerations for how to assess an AI algorithm might apply, of course, to us as well. You might want to adopt this a little bit for your specific use case. There's two resources here. One is from CLIA around what was the prior, what is the current definition of LDTs, especially under the regulatory updates. And then a quick pointer to an article from Karen Titus, I think published by now in the CAP Today, as well as several of the CAP resources. Excellent work. She has interviewed many people and tried to summarize some of the uncertainty and some of the issues that come with this rule. In this paper here, I wanted to, of course, give a pointer to the paper itself, but wanted to specifically point out the table S1. It was too big to squeeze into a PowerPoint slide. But if you're interested, or if you write a paper on AI and you want to systematically assess biases, the supplemental table 1 here is really noteworthy and a great resource to have. Similarly, Eric Dunkevich from the Washington University in St. Louis published with his group a paper in JMD about an analytical validation of a multi-target stool RNA test. And why I wanted to point this out is that it is a phenomenal demonstration of how to do an analytical validation. So great paper, didn't put any figures in, but the outline of how to do this is really phenomenal. So thanks a lot. Two other quick resources. I think this is coming from one, and then there's another, I think 2 or 3, which is from NIST, which in 1819 they put out this so-called real-time public safety data challenges and possible future states. For example, determining approaches to data access control, as well as, you know, data sharing, etc. And notably, in July, there's a series all packed into one document that is outlining the topics here that might be of interest to many of you. These are, again, very systematic approaches to enable the topics that NIST is here proposing. So a series of documents with great resources. Another resource you see here, this is from 2018, but there will be a paper in a second that cross-referenced this, and the paper was sent to us, but I found this resource and I thought that for some of you who work in the drug development business might be very interested. So this is from the, you know, HHS. It was submitted to Erin Rubens, a final report on the general cost by biomarker category, as well as a breakdown of the cost ranges for, you know, identification, development of the assay, analytic validity, and clinical validity for predictive versus surrogate endpoints. And those are, of course, two of the heaviest or most complicated biomarkers to work with. So I thought that was a tremendous resource, at least if you need to cite something that might be helpful. And again, there's a paper coming in a second where they provided an update to this study. So I wanted to share this as a resource though. There was also the International Council for Harmonization of Technical Requirements, Pharmaceuticals for Human Use, ICH. They release, I think, very complicated and very helpful guidances that are often referred, cross-referenced, or directly copied by the agency. And in this case here is about pharmacoepidemiologic studies that utilize real-world data. And there's an exact framework provided. I don't think we're quite there in the device world yet. But again, in terms of using this as an additional resource, want to provide this. In terms of pharmacogenomics, this is a very phenomenal multi-society position statement. So if you see that this is DPYD genotyping recommendation, and this is a joint consensus statement coming from all these different societies that work on this, quite phenomenal. I only had the pre-proof. There's actually table 2 shows tier 1, table 3 shows tier 2, DPYD variants with exact coordinates, reference position, as well as the consensus statements around these. So I believe that that's very helpful. And then as an additional resource from Dr. Priya Singh, a series, it's a website as well as some YouTube videos on how to perform and conduct a systematic review, which might be of interest. I watched the video series. It's actually very, very clearly explained. And if you haven't done systematic reviews, that might be helpful. And then I think Eric Topol on his site has recently interviewed Faisal Mahmood, and he gave a podcast, so quick pointer to this. I will go over the publications really quick. Again, these are just sort of quick pointers to the papers. The numbers seems to surmount to even more. We will share, of course, all these publications with you on the website. I just literally try to say one sentence per thing so we have some time to discuss. Several algorithms, in this case here, a foundation model, which seems to be a trend. You will see at least 5 or 6 publications of foundation models here. And the main point in those is, of course, is this, with or without large language models, and what is actually built. And here is a combination of tools applied. I think that is currently available in preprint. Another preprint here from the group around Dr. Sun, illustrating here how to accelerate clinical evidence synthesis. So the combination of various sources of literature as well as data aggregation and literature screening using LLMs. In this paper here by Paul Hoffman and group, we last month mentioned that there's sort of a trend that, cancers of unknown primary should be classified based on molecular typing. And here is how to change tumor naming and, the molecular classification. So this is a continued theme, and, having this come out in JCO is a clear indication that there is sort of, especially when it comes to treatment selection, a trend to consider that. This study, phenomenal by Mackenzie Evangelist, and of course many others, is continuing her work that was illustrating how prevalent biomarker testing in non-small cell lung cancer is. And the highlighted section here shows that 54% of patients received testing for at least the main elements. And of course, with additional therapy being offered in patients with biomarker positive tumors, however, 16% still received no molecular testing at all. Now, this is a huge improvement over, I believe, 50% about 4 or 5 years ago. But you see here the breakdown of what else is considered for the different, for example, EGFR mutant versus not tumors. But again, a quick pointer here. A group around Dr. Moseli and Matteo and Andre have updated the ESMO guideline for the 2024 NGS treatment. And keep in mind, this is, in this case, is a special article, and there's recommendations about tumor agnostic genomic markers, etc. And this is a continuation of a prior work that I also screenshotted here, I believe, from 2020. And that's the same group. I'm pointing this out because there's a separate paper, almost the same topic, but more for the reporting. So not the testing recommendations, but the reporting. We get to that in a second. This paper here, deep learning models illustrating a high relevance of determining a BRAF positive atypia of unknown, undetermined significance identified as malignant by AI models, wanted to portray this, that this is a retrospective, prospective multi-center study. So I think retrospective assessment, but prospective enrollment was the design here. This paper, Nature Communications, by Ali Bashashadi's group, illustrating the identification of a new subset of endometrial cancers, including, a sampling approach. In this paper here, 10,478 cancer genomes identified, and you see here the effect size in terms of how many samples do you need to test to identify key drivers in these different cancer types. This is the paper I mentioned earlier about the low-cost battery-driven device. So this was a randomized non-inferiority trial published in Nature Medicine, and this was from the prior WHO update. The Lancet, in this case, a commission's article, portrays in epidemiologic terms that there is apparently a surge in cases, largely related to increase in androgen deprivation therapy and effectiveness of so-called next-generation anti-androgen therapy, therefore leading to more therapies as well as more cases with relapsed disease, illustrated here. Whether that's true or not remains to be determined, but that provides a clear vector that a larger scale epidemiologic studies in pancreatic cancer as well as the diagnosis of prostate cancer remains relevant. This paper by Andrew Janovchik, NPJ, so Nature subjournal, is illustrating a so-called cohort finder, which is an open source tool for digital path image cohort assembly. So you can take multiple data sets and assemble them, including high dimensional QC. Phenomenal paper, and certainly one of the approaches you will see several others are emerging as well. In this paper here by Ananyana Chowdhury and Catherine West, this is muscle invasive bladder cancer, including digital spatial profiling. You see here the concept map, also a phenomenal paper. Thank you for sharing. Here's a paper by Mariano de Socaras, good friend and participant, of course, here. I don't see him on today, but of PIcc, where they report a 3-year clinical experience using an AI identification algorithm for prostate cancer. And I think it's a very realistic study. This is 122,000 slides, over 9,000 cases over 3 years. And in large oversimplification, they've done a lot. They ran the prostate AI algorithm on all cases, basically as a QC measure afterwards. And the overall impact was a factor of 1.8%, which I leave that to you to decide whether it's the same for your practice. But I think it illustrates in great detail what that means. And 2% out of 122,000 slides is not an insignificant amount of numbers. I have to go back and look what the patient numbers are, but this is a pre-pub from Thomas Fuchs about benchmark embedding for aggregation models in CompPath. And there's a great timeline on how this field of spatially aware or spatially unaware elements or functional tools has evolved and, of course, continues to evolve. This paper from Bettina Sobotka and Holger Moch's group and Hendrik Kulzer is a illustration of an immune phenotype-genotype association, in this case in clear cell renal cell carcinoma. Interesting, the analysis between primary as well as metastatic tumors and co-localization analysis. Related to the epidemiologic papers that we showed earlier is a paper by Sebastian Schneeweiss from Harvard, which is an enhancing external control arm analysis paper as a tutorial. And while the quality here in my blow-up is not quite as good, when you see the paper on the website, this is illustrating exactly how to, for example, create control arms if it's in randomized settings or in underpowered settings. Very, very helpful. In this paper here that was published at the end of May, the authors of a routine whole genome sequencing for children define certain impacts. Why I wanted to share this quickly is that whether a test or a tool has diagnostic impact or aided in a diagnosis or had therapeutic impact or changes management was classified here. And you see here the breakdown in this particular setting of applying whole genome sequencing. This is a trial using a retrospective analysis of an immune signature in ductal carcinoma in situ, and high tumor infiltrating lymphocyte density was associated with increased or higher risk for recurrence. This is an interesting systematic comparison of sequencing-based spatial transcriptomic methods. You see here the different elements compared in a confusion matrix. If you're into the sequencing spatial transcriptomics, this is a must-go paper. AI in real-world generalization. There's a very interesting part that certain AI models leverage demographic shortcuts, in this case for disease classification. So this reminded me of some of the USMLE questions that were identified as potentially undergoing racial bias that were eliminated a few years ago. And based on this paper, this seems to be similar. But there's, of course, a lot of other messages in the paper. I just wanted to highlight that out. There's a great paper by Renato Umeton from Dana-Farber Cancer Institute and the group using ChatGPT for digital pathology research. And they provide a lot of resourceful tables about what data sets have been driving these models and how and what to use those for. So a great pointer if you want to work in that space. It's a great review on early cancer screening and surveillance. There's another one coming up in a second. Haifeng Poon and group have provided this whole slide foundation model, I think called Prof GigaPath, and it's an open weight foundation model that achieves a state-of-the-art performance. I believe there's 17 tasks, probably a couple of more, and 30,000 patients, etc., in 31 different tumor tissues. In this paper here by Dr. Koskun, we talk about, or they talk about multiplex bioimaging of single-cell spatial profiles. This is just a concept map. It goes deep into tumor microenvironment profiling, etc. As promised earlier about the cost of drug development during the resource section here, let me just go back real quick if I find it. This paper here, 2018, was illustrating the drug cost. So you see, for example, the total for surrogate endpoints is 23 million, or predictive biomarkers here, 15.7. And in this recent paper by Dr. Sommers, collecting data, costs of drug development, in this case in the US from 2000 to 2018, in the subcategory of oncology, you see here are the different phases of the cost that's annotated, including the FDA review. And you can read in the methods exactly how they did this. Is broken down, in this case, also across the different other groups. I had to cut the table because it goes across all the different disciplines. So there's a cross-link here. And of course, if you're a drug developer, that might be helpful. This is the other ESMO paper that I just mentioned, in this case on clinical reporting. This is published in the Annals of Oncology. And you see here's certain elements that I believe are not common standards for reporting in the field, but they're definitely now on the ESMO recommendations part. An interesting paper here by Dr. Wong from Vanessa Weir et al. Is how to do, or how to survey skin tone assessment in prospective research. You see here is prospective research study recommendations in terms of ranging from underlying conditions, ambient lighting, and all the elements to ensure that if you record skin tone, it might be helpful to consider that. In this paper here, a machine learning model for complete response to radiation and rectal cancer. You see here's a element with quite high sensitivity and specificity. This is complete resection non-small cell lung cancer and survival depending on the specific substage. Here's a multimodal breast cancer predictor by Dr. Mann et al. This is a great paper by Thomas Westerling Bowie and the group. I believe most of the folks are at Mayo, where they present an ecosystem, and if they give pathologists the opportunity to train multiple models, they mention it and they call it a democratization to support pathologists with varying levels of experience, that can break down entry barriers for AI. Published, I think, in the archives, and very interesting approach to enable pathologists even without that. The paper on fibroblast activation protein and some of the compounds available that might be a new target. So look out for FAPI, which is the inhibitor of that. This is a pre-proof from Hamid's group, a great group that they provided something called SPLICE, which is streamlining digital pathology image processing. Again, a great tool to work on this. This is from Sylvia Thun from Berlin, an article about interoperability landscape. And here are some of the coding frameworks in the semantic as well as in the syntactic framework. So you see DICOM here, for example, versus ICD-10, etc. So great overview. A few more papers. This is tumor-associated macrophages, which might be relevant to some of you who study the microenvironment. This is a paper illustrating the limitations of large language models by Daniel Rickards' group from Munich, illustrating that, depending on how you define a certain conversation, which was done here in an evaluation approach, whether that is better or worse than doctors. And, for now, at least, it seems that doctors still have some power, but that will probably soon be improved. Quick outline here. This is a real-world evidence of overall survival in non-small cell lung cancer that was shared, and comprehensive genomic profiling was associated with improved survival. Here's a similar study to the one by Mariano, in this case for prostate and breast cancer. I think in this case, diagnosis and grading, a retrospective, a much smaller cohort. In this case, I think from a Japanese group, Dr. Fukuoka, and you see here the positives and negatives, but also wanted to share this. Here's 3 publications that we haven't featured before by Faisal's group, all available, of course, on the website once they're out. Thomas Fuchs's foundation model published in Nature Medicine, including cancer, benign and unknown settings, great figures, great overview. Please take a look. In this case, another paper in NPJ Precision Oncology from Ali Bashashadi's group. So you see there's some people very, very active. This is a generalizable AI model for multi-center histopathology image classification. And if you look at the numbers, that's quite impressive. So including Fourier transformation. In this paper here, Dr. Lu's group in NPJ Digital Medicine, they examined a 2017 New England Journal of Medicine image challenge and how GPT models can help and or not. So incorrect classification, etc., is mentioned here. This is an ethics paper by 2 doctors that performed a systematic meta review of 53 papers that illustrate and emphasize that fairness, bias, and some of the other components here, including privacy, are some of the main concerns. Finally, the last couple of papers, this is a review type article on challenges and opportunities in multi-omics integration. And here is a quick depiction of the multi-omics approach that went all the way to microbiome and level of understanding or uncertainty. One paper was shared that I thought was, we're switching to the final 2 or 3 papers that are somewhat funny. This is a ketogenic diet. There's some image analysis in there, so maybe that's why it was shared. This is a great paper illustrating the so-called Picasso skepticism on computer science. Here's a OpenAI generated Picasso cubism-like MRI image of the brain. And the interesting thing is that the group argues that humans should stay in the loop and call the approach machines in the loop approach. I don't know, more sort of tongue-in-cheek, I guess. One group published, and I think Brittany was mentioning earlier, the idea about HER2 quantification and that there's a lot of concerns around it. And this group, I think around a research group from AstraZeneca, Anj Kapil is the first author, has illustrated, and I thought this figure I definitely wanted to show is on the bottom here is the amount of response to trastuzumab deruxtecan. Well, this is the ADT for HER2, including HER2 low, and here you see the different scores. So clearly there's no like immediate pattern, but the group deducted through a spatial proximity score that apparently the distribution might matter, and they argue for a more continuous scoring than these 4 categories. Now, this is published in scientific reports. The evidence is great. I just wanted to share that this is probably important for us to cite in this HER2 study, but also in general, an interesting approach if there's not a direct approach in terms of the existing classification, how to overcome that and argue for something there. Finally, there's an interesting expert voice paper by Eric Topol around the eradication of medical diagnostic errors. I let you read that and take your own position on that. And then, next meeting is, of course, at the last Wednesday of August. And 2 other courses that some colleagues have recommended, and I wanted to remind everyone, please do share this with us. We can definitely feature it. Is the Ohio State University has a meeting here September 12th and 14th, and API is organizing a fall meeting September 15th and 17th. So that's the quick overview over all the things that everyone has shared. Thank you very much, of course. And I want to open it up for a few minutes of questions or comments. I think there's some parts in the chat that I need to catch up with, but while that's going on, let's open it up for comments, questions, or anything.
Outstanding job, Joe, as usual. Really impressive. Thanks for the community for this kind of oversight of all these and availability of awareness of all these resources. So I only ask that they're on the website, right? It will be available.
Yes,
Nice.
I think we discussed internally with Noor that if we drop all these PDFs,
Yeah,
people will not. I mean,
it'll crash. No,
we will have them all on the website. So
Yeah.
I just wanted to go over this real quick because I think it works well. A lot of people share all kinds of resources, which is fantastic.
Yeah.
But we thought that for the recording, it might be easier to just sit there and watch, and then you get all the info later. All right. Thanks, Keith. Great seeing everyone. Hope to see you soon, and please keep moving and pushing the field forward.
Bye, everyone.
thank you, Joe. Thank you, everybody.
Thank you.